Okay, it's time we start on our section having to do with embryology. Before we start looking at the developmental processes that go on to form the anatomy, we're going to talk about the different kinds of eggs that are produced by different members of the chordate phylum. The term microlecithal, micro means little, lecithal refers to a yolk material that's found in the egg. So those that have very little yolk material to contribute to the developing embryo in the egg is microlecithal. Next would be mesolecithal. It means it has a moderate amount of yolk. And then macrolecithal obviously means it has a large yolk, like a bird egg yolk. So these eggs can vary in the amount of yolk nutrient material present in that first cell, that embryonic egg cell. So these differ in size, and microlecithals usually are nearly invisible. Can't be seen or just barely at the edge of what's visible without the aid of a microscope. Mesolecithal are easily seen in things like fish eggs and frog eggs. We see fair amounts of yolk in them, so they're easy to see those eggs. Then finally macrolecithal, very large eggs like we see in turtle eggs and bird eggs and so on. So those are the conditions we're talking about: microlecithal, mesolecithal, and macrolecithal. In addition to that, the distribution of the yolk within those is not going to be the same between the different types. Generally, in a microlecithal egg, there's so little nutrient in there, we consider it to be equally distributed within the egg, so it's considered isolecithal. Isolecithal, there's a small amount of yolk in there. If there's a fair amount of yolk in there, it has to be located usually at one end of the egg, or one pole, what we sometimes say. So rather than being isolecithal, these are telolecithal. A little less telolecithal means that it's polarized. There's an animal pole. It contains the nucleus, and then there's a vegetal pole. They contain the yolk nutrient. Now let me kind of illustrate what that would look like. If it's just a very, very tiny egg and there's just a single nucleus in there, any yolk material that would be present, indicated with a red color here, would be kind of uniformly spread through that very tiny egg. If it's a mesolecithal egg, larger kind of egg, it's going to have a tiny nucleus, usually at just one end of the egg here, and then the yolk nutrient material is distributed mainly to the opposite pole of the egg. So all this is yolked down here. This area up here around the nucleus is not yolked. So we have a polarity going on there. And in a macrolecithal egg, things are exaggerated to even a greater extreme, and I can't even put the whole egg on the board here, so I'll just indicate a little bit of the egg. Here's a nucleus right here. And really all the rest of that would have yolk material. The yolk nutrient is distributed throughout the rest. So only a small area around the nucleus would not be yolked. Very, very tiny a microscopic kind of area around that, would not be crowded with yolk. This affects how the cells divide when you get to the first division of the egg after it's been fertilized. This first example, the isolecithal, microlecithal egg, when it divides, it's going to become two equal-sized cells. So it's going to have what we call equal holoblastic cleavage. Cleavage event is when the cells divide, that's splitting event. So when it splits, cleavage event, you're going to have two equal sized cells. They're not going to be one large cell and one small cell or anything of that sort. The mesolecithal egg, when it starts to divide, the division occurs between the two nuclei that form. You get a cleavage furrow to start developing there, but that cleavage furrow is very, very slow to move through that yolk. And that nucleus will divide again once or twice before that first cleavage furrow divides the entire egg cell. So we call that unequal, but still holoblastic. Unequal holoblastic. Cleavage. Unequal holoblastic cleavage. And we'll illustrate that when we talk about frog egg development. So we'll see how it starts to split the yolk, and then another split starts to form before the first split goes all the way through, and then another, and so on. So we'll end up with a lot of tiny cells that are dividing rapidly up here, and very few cells down in the yolked portion. They're larger. When you get to something the size of a bird egg, where you have a large yolk, when the nucleus divides, you start to form a cleavage furrow, and it never really penetrates through all this yolk material down here. So you get just a whole bunch of dividing up of cells right here in this one small area above all that yolked material down below. So you end up with a lot of tiny cells up here, but no division of the yolk down below. And we call that meroblastic cleavage. Meroblastic cleavage. So we see that in reptiles and birds, and you'll see it also in some sharks that have large yolks in there, and probably the coelacanth as well.